Okay, great. Hi, good evening, everyone. Thank you for joining us for the webinar. We'll just wait for another minute or so for a few more attendees to join, and then we'll start with the discussion. Yeah. Okay, great. So we'll start with a little bit about settings. I'm just setting some context here about the program and the Thought Lab and the webinar we're all here in for. So this session is brought to you by the Lodha Genius Ashoka University program. And the Lodha Genius Ashoka University program is a joint collaboration between the Lodha Foundation and Ashoka University. And it was started with the idea to nurture young minds all across India and to give them access to research labs and experts from all across the globe so that they learn from the best. They come to Ashoka University, get access to research labs and all the resources over here. And once they go back to their homes and to their schools, they work on solving very pressing issues in the society. And the idea is also for nation building altogether and for these students to just give back to the society where things actually matter. So, as part of this program, we started a webinar series called the Lodha Genius Thought Lab. And the idea for this webinar is to have very meaningful and engaging discussions. But these are also the topics we choose are also the topics that are not usually spoken of. So the idea is also to raise awareness amongst the general citizenry and amongst students as well, for them to be equipped with certain knowledge and to also be inspired by our amazing speakers and experts that we invite. So today's discussion is a very interesting one. And the guest speakers that we have today are Uma Ramakrishnan and Prasenjeet Yadav. Uma Ramakrishnan is a professor of molecular ecology at NCBS, Bangalore. Her research is deeply rooted in India's biodiversity, and her lab also uses population genetics, landscape genomics, and evolutionary biology to understand how human pressures shape wildlife populations, right? And she's made very interesting, great contributions to the field of tiger conservation as well. The second speaker we have here today is Prasenjeet Yadav. Prasenjeet Yadav is a molecular ecologist turned National Geographic photographer and explorer. He holds a master's degree in molecular biology, and he has worked for several years at NCBS in Bangalore, but now he combines his scientific training with photography to tell powerful conservation stories. He focuses on underrepresented species and landscapes, collaborating with researchers, policymakers, conservationists, and his work has highlighted issues like climate change in the Himalayas and ecosystem impacts across India. So we welcome the both of you here. This session will also be moderated by Professor K. Vijay Raghavan. Vijay is the chair for the Science Advisory Council at Ashoka University. Vijay is also the former principal scientific advisor to the government of India, and Vijay has been that person who's really pushed for the advancement of sciences in India throughout the years, and he's contributed so much to the field. So any introduction, I guess, is it falls short of the work and the dedication Vijay has overall. So I will just some housekeeping rules for all the attendees over here. We do we'll have this discussion for 35 minutes. And if at any point of time you have any questions, please feel free to drop them in the Q&A box and we can take them up after the discussion. Because after 35 minutes, we open the room for audience Q&A. And yeah, so I'm handing over the session to Vijay now. Vijay, the floor is all yours, and you can lead the session.
Thank you very much, Utsav, and a warm welcome to Uma and Prasenjeet. This is really one of the most exciting thought labs I'm sure that you'll go to see and hear about. Uh, Growing up, before the internet age, the magazine which one would go to in libraries and read cover to cover was the National Geographic. And in the age of the digital age, many people read it online, but you don't get the same feel. I would urge you to go and get this October issue. Oop, my background is blurred, but you can't see it too well. Get the October issue of National Geographic. On the cover is a story which Prasenjeet has photographed and written, and it's perhaps the first time that someone from India has featured a cover story like this in National Geographic. So, warm congratulations to Prasenjeet. Before I pass on to him and then to Uma, I'd like to point out a small factlet. 10,000 years ago on this planet, humans and uh, our livestock consisted of 0.1% of vertebrate biomass. Amongst all the vertebrates on the planet, we and our livestock, cows and pets and so on, constituted only 0.1% of vertebrate biomass. Today, humans, our pets, and our livestock constitute 98% of, vertebrate biomass. So today, we, in a sense, rule the planet. We not only have populated it densely, but we control everything on this planet. And so it's not just when we look at the world around us, it's not just to understand it, but we are the stewards of this planet now. And so we are responsible for what happens. Look at that also from another perspective. Our understanding through physics, chemistry, and biology of life and everything else around us has improved tremendously. And we can make a link between all of this beautifully. Today's story about the black tigers of Siripal and tiger conservation is an extraordinary example how from the most fundamental of molecular biology to survival of animals in our ecosystem, the link is absolutely unbroken. And that story is a very, very exciting story. And you'll hear about it from the authors of that story, Prasenjeet and then Uma. So without any further ado, over to Prasenjeet.
Thank you. Thank you so much, Vijay, and thank you everyone for joining us here this evening. Let me try and see if I can share my screen. Give me just a moment. Yeah. Very cool. In Prasenjeet, I essentially am a photographer, filmmaker, and I also write at times. And I do this for National Geographic. And in the last 13 years, they have tasked me with some unusual, hard, weird stories. I've worked on bats in South America, I've worked on mountain goats in Himalaya and Mongolia, and I've worked on tiny frogs and snakes across Western Ghats. Uh, but last year, 2 years ago, I was tasked on photographing this one crazy tiger, uh, which looks very unique. And 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 I'm not saying that in a poetic sense. It is very unique. It's a black tiger. Science calls it pseudo melanistic tigers, and Uma will tell you more about it. But this particular individual was called T12 or tiger number 12. And I got obsessed with him. My task was to photograph this tiger, get a ultimate picture of him. And first, I went there with the hope that I'll be able to get it in like a month. It turned out to be 4 months and two different trips. These tigers are found in this one forest called as Simlipal, which is located in Odisha, India. And as you can see, Odisha is this humongous and a beautiful forest. It's almost 3,000 square kilometers in size, and it's sitting on a tabletop plateau, which is almost 1,000 meters above. And in this forest today, there are approximately 30 known tigers, out of which half of them are to be identified as black or pseudomelanistic, as they are called. So when I went there, I went with some experience. I worked on snow leopards in the Indian Himalaya, and it is one of the hardest species to photograph. So I went there with this experience and I thought that I worked on snow leopards. I should be able to get these tigers. It shouldn't be that hard. Turns out it was much more harder. Once I got there, I realized the knowledge of where they move, when they move, was very limited. So I did what I'm really good at. I sniffed trees. I did this for weeks. Tigers communicate with each other by spraying on these trees, and if you are willing to get your nose involved, you can learn a lot about their movement. So by the end of a few weeks, I had deployed 10 of my good cameras or camera traps to be able to photograph these tigers. And by the end of the month, I had just one photo. And this wasn't a good photo either. I wasn't happy with it, but it kind of gave me a sense where they move and when they move. And this is just to show the sheer size of that tiger, just to put things in context. I'm 5'10" in length and height, and you can see side by side at the same place how humongous that tiger is. Uh, T12 and tigers which were born almost 10, 12 years ago are extremely shy because a decade and a half ago, Simlipal was dealing with a lot of social unrest as well as some kind of hunting pressures. And all these individuals learned to survive just by being extremely skittish and shy. So if the tiger would hear humans or see a human or smell human, they would just leave the trail. And that is what made photographing T12 extremely, extremely tricky and hard, unlike any other tigers. And not all tigers there were shy. I mean, this is not the tiger. This is me testing my camera. And I left, and within an hour, there was a black tiger in this same spot. But this tigress is born almost 2.5, 3 years ago, and she is not camera shy at all, as you can see. Uh, because now Simlipal is a beautifully managed park with a lot of protection, and the tigers which are born in recent past feel much more safer in this landscape. Uh, the first trip I made was supposed to get the picture, but almost after 65 days, I did not get picture. And the monsoon arrived, and I had to leave that place just because monsoon shuts it off entirely. But National Geographic created a small video of my first trip being there. And let me play that for you right now. Tiger spray smells very peculiar. It actually smells a lot like the basmati rice that we get here in India. And this one is fairly strong. So that means that probably one of the tiger has moved from here not too long ago. Simlipal Tiger Reserve is 2,700 square kilometers, which is humongous. And within that, there are known 30 tigers, out of which probably 15 are black or pseudomelanistic, as we call them. So I would say photographing them is harder than finding a needle in a haystack. It could take me anywhere between 2 to 6 hours to set up one of these cameras. I found the trails which were most used by tigers, and I deployed my best 10 cameras on those best 10 trails. We have checked 6 cameras since morning, and none of them have tigers in it. This is the last one for the day, and I don't know. Nah, nothing. Nothing. I spent last half an hour trying to figure out why my camera trigger is not working. And this is what I found. These little guys, they went inside, and one of them is sitting right on the sensor. And then I had this one camera which was just spare. And I was like, I'll deploy it somewhere close to the base camp that I live in. We left, and within an hour and a half, we got our first picture of a melan's tiger. But I still haven't got the picture that is there in my mind. And that's the reason I'm going back again. So I went back again after the monsoon, and I spent another 60 days. And then on the last second day, before I was exiting the park, Tiger's Free, I got this picture, and it made it on the cover of this magazine, which is crazy and one of the biggest and craziest experiences of my life. But the reason this picture made it to the cover is not because these tigers are unique or this photo is out of the world. Along with that, it also made it to the cover because the story that it represents is much more than a beautiful picture. And it represents science and conservation efforts, science done by NCBS and Dr. Ruma, and conservation efforts by Odisha and Maharashtra Forest Department. And I would let Uma speak a bit more about the science part of it, why these tigers are amazing and crazy looking. Over to you, Uma.
Thanks, Prasen, and thanks everyone for being here. It's really fun to be part of this conversation. So, the amazing thing about evolution is uh, it's all around us, right? That's what we hear, but seeing evolution is a little hard sometimes. For example, we can imagine how coronavirus, well, we saw how coronavirus evolved. There were so many different strains. We were talking about it all the time, the Delta strain, etc., the Delta variant. Uh, but to think that tigers can evolve or change seems really odd, right? But exactly as Vijay said, the fact that these animals and their habitats have changed so dramatically in the last 200 years. The forest cover or green cover in India has gone from 70% to about 10 or 15% now. And you can imagine then living in this large, vast, green space, which is shrinking continuously. And so finally, if you're a tiger, for example, you're stuck on an island. And when you're stuck on an island with very few neighbors, right? And very few individuals in your population, what happens is a phenomenon called genetic drift, that by chance, certain mutations, certain genetic variants increase in frequency, just by chance. And so that's what happened in the case of the Simlipal tigers. We were lucky that we were able to capture this evolution in action. Basically, human effects, fragmented tiger populations resulted in isolation of certain populations. And because they were really small, they went through these bottlenecks. There was also inbreeding, which resulted in increase in specific genetic variants. And again, we were very lucky that in the case of the black tiger or the pseudomelanistic tiger, we were able to figure out what was the spelling mistake, what was the mutation, what was the genetic variant that made it look this way. And it was simple. It was what's called a Mendelian trait, that is a single point mutation or a spelling mistake. And if you have two of those spelling mistakes, one from your mother and one from your father, then you look black. Whereas if you have just one, you don't look black, you look like a normal tiger, and so on. So there was a lot of luck involved, and it could have been that this was a very complicated trait, maybe like heart disease or diabetes, where there are many, many genes involved. But in this case, because it was so simple, we were able to find it, and we were able to show that because of isolation, there has been evolution in these populations, meaning change in allele frequencies, change in genetic variants, which has resulted in changes in phenotypes, making the black tiger, which is otherwise rare or absent from outside of Simlipal, common only here. So anyway, so that's kind of what we found, and it took a lot of genetics and field sampling, collecting samples from the field, bringing them back to the lab, analyzing the DNA, and doing a lot of calculations to see how could we explain what we saw. But yeah, so that's kind of the story of how we found out about this population and what was going on. And we also then suggested that we model the population into the future, and we showed that given this isolation, unless there was inflow, uh, over time, this black mutation or this variant would get fixed. Now, we haven't shown or we're not suggesting that the black mutation per se is bad for the survival of the tigers, but it is associated with an inbred population and indicative of higher genetic load or more, potentially bad mutations which occur when there is inbreeding. I hope that's not too complicated. And so at that point, since we suggested that through these simulations that these mutations may get fixed, there was some interest in genetic supplementation or introducing individuals which would bring in variation, new variation, as well as offset this increase in frequency of the black mutation. And we were very lucky that we were able to collaborate with and work with the forest departments of both Odisha and Maharashtra. And they, of course, implemented this translocation movement of individuals. So ideally, before the forests were fragmented, the tigers were moving on their own, right? But now, because of fragmentation, there's too many blocks for them in the way, and they can't move on their own. So humans who caused the fragmentation initially brought individuals, and now these two tigresses have kind of settled down in their new home, and we'll wait to see what happens in the future.
Thank you. Thank you, Uma. And Prasenjeet, that was very nice and very, very impressive. Uma, you mentioned that the pseudomelanistic tiger is because of a mutation in a particular gene, and when the cub carries both mutant copies, then it is pseudomelanistic. Now, what is the nature of this gene? What does it encode? Do you have some idea? And how did you discover that?
Yes. So that's really cool. I mean, basically, this is a gene called TACPEP. It's a transmembrane aminopeptidase. But what's interesting is it's involved, though we do not know exactly how, it is involved in pattern formation. So there was earlier work with cats where they actually showed that TACPEP and mutations in TACPEP were involved in cats going from regular patterning of stripes, like the tabby pattern of cats, to a world or like a mixed up pattern. So basically, for example, in the case of a normal tiger, TACPEP would be involved in saying start black stripe, stop black stripe, start black stripe, stop black stripe, so that you have a regular patterning of the stripes. But with a mutant TACPEP, which is non-functional, there's no start, stop, and regularity. And so the black just goes all over the place. You might have noticed if one looks at various individuals, there's also a variation in the amount of black. So we don't really know why that is. But because of the paper which was already published on cats and also a similar pattern disruption in cheetahs, which don't have stripes, but their pattern is also disrupted when they have mutant TACPEP. We had a candidate, so we could just quickly zoom in. Otherwise, finding the genetic basis of a phenotype in the wild would have been really, really formidable.
So, Uma, because this is a isolated inbred population, you have many black tigers because those which are carriers and not black, right? When they mate with another carrier or with a black tiger, you will get black pseudomelanistic progeny. And if they mate with each other, you'll get more and more pseudomelanistic progeny. Now, in many ways, this pseudomelanistic tiger is also a visual indicator of inbreeding, and therefore lots of other genes are also homozygous, or both copies are similar, and therefore the what will happen to the tiger? Will it be stronger or weaker because of this inbreeding?
So that's something. What you talked about, Vijay, is what's called inbreeding depression. So, inbreeding is mating between relatives. And it so happens that relatives have a common ancestry, a common past. So relatives share many deleterious or potentially bad or damaging mutations. Now, the thing is, in large populations, if a mutation is very damaging, and if it is dominant, selection would clean it out, right? Those individuals would die, and then it would no longer be in the population. But these recessives, or the kinds where you need two copies, would hang around, and all of us actually have these potentially bad mutations. What happens in inbred populations is, because there's mating between relatives, and because relatives often share these bad mutations but have them in a single copy, they come together in the offspring. And so inbreeding depression is the consequence of the coming together of these bad mutations on the offspring in terms of fitness or their survival. So outbreeding, or mating with very different lineages or ancestries, actually results in higher vigor, or heterosis, or hybrid vigor as it is sometimes called. But inbreeding results in poorer survival, most often in the wild, seen as poorer juvenile survival, shorter lifespan, or lower reproductive success. So this has been documented extensively in breeding depression in human societies, in experimental animals like mice and Drosophila and so on, but also in wild populations, and how inbreeding affects the future. And Darwin actually talked about inbreeding and inbreeding depression. So it's something which is a kind of an old topic, and has been explored quite a bit.
Very exciting. And Prasenjeet, can you tell us about how Uma and you worked with the forest department, or Uma worked with the forest department to have a tiger move from other sanctuaries? What did it involve? How was the tiger anesthetized, brought over, and was it successful?
Oh, that's a great question. Yes, I think Uma's paper had advised that translocation would be an ideal solution. And I think Odisha Forest Department took it seriously. They there were right set of officers who were like, okay, although inbreeding depression is not there, we know that it might be there someday. Why to wait for something like that to happen? Now, ideal scenario would be that tigers move in on their own. And of course, the forest department is working on restoring corridors so that the tigers can move in on their own. But that might take much longer. It's not something that can happen overnight. But what could happen overnight is to physically lift tigers and bring them here. So what was done, from what I've discussed with Uma and with forest officers, that there were two things that worked out. Uma's research also showed that Tadovandhari Tiger Reserve has very diverse set of DNA and very limited genetic overlap, negative overlap with Simlipal. And at the same time, Simlipal Forest Department and Odisha Forest Department had great relationships with Tadoba and Maharashtra. So all these things came together. And then to translocate a tiger, it's not just that two states converse and they are like, okay, we want tiger. Oh, you can take tiger. No, there are a bunch of permissions that had to be brought in place from the central government, from MOEFCC, from NTCA. So it took a while for them to get all those permissions. When permissions were in place, then I got lucky that they allowed me to be part of the operation. And as part of that operation, we all drove to Tadoba, and then we were like, okay, things are ready. And Odisha's team of 25 people, Maharashtra Forest Department's team of 25, 30 people, all of them were ready to take tigers, but then the tigers had to be caught as well. And many of these tigers are so shy and hard to find. And then to be able to find them, and if you find them, to all the resources around you at the same time, and then the vets has to be there, and then in the jungle finding that tiger, and then darting it, and the dart has to hit the right place. Many times they saw the tigress, but the tigress was sitting in a way that there was no proper shot to be taken. So it took weeks before they could tranquilize one of the female tigress. And then the thing is, once you tranquilize a tiger, it doesn't get sedated at the same place immediately. It runs. And sometimes it runs for 50 meters, sometimes it runs for 200, 300 meters. In that forest, you don't know how many other tigers are there. You don't know where exactly this tiger is sedated. So then there's that risky business of going and locating that tiger on foot by 20, 25 people and then checking if the tiger is actually sedated or not. Because what if it is just drowsy and wakes up and says hello? Uh, that's it, and it can make a huge damage. So fortunately, they managed to tranquilize, sedate, capture one of the tigers. Then it was put into a big truck, and it's the first time that Maharashtra and Odisha Forest Department drove a tiger in a truck for 1,100 kilometers. That's never been done before. 1,150 kilometers over cities and villages, and then it was brought to Simlipal. Now you can't just release it in Simlipal because the managers have to make sure that animal is moved for like 28, 24, 27 hours, so it is doing fine. So what they had created is they had created this huge enclosure of 1 hectare inside the forest where they released the tiger first, and it took them months to build it because it has to be big enough and strong enough that an animal like tiger can't escape from it. So they kept the tiger there for a week, 8 days, till the tiger acclimatized, and then eventually they opened the gates and the tigress moved out into the forest. They repeated the same exercise one more time in early November last year, and 2 female tigresses were moved to Sindipal. It was an incredibly huge, but to be honest, uneventful uh, process. Uneventful is very good thing when you're catching a tiger. You don't want special events happening. So it was done so well that nothing out of it was crazy.
And was the move successful? Are there progeny now, or is it too early?
No, it's a good question. So first tigress, which was removed, is right now living on the periphery of the park. So she's settled around this place called as Kuldia, which is literally right outside the Simlipal Tiger Reserve. Another female which was brought in decided to scoot out, and she went to Jharkhand and some other state, and they eventually managed to capture her from West Bengal and bring her back, and now she's settled inside Simlipal. And at some point, the forest officers even saw her meeting with T12, our guy, the big male. So we don't know, in while many times they mate and progenies are not born immediately, we don't have records of if there are miscarriages and all of that. But the thing is that the tigers, both the female tigresses, are settled in and around Simlipal. That itself is a success, shows the success of this event, of this process done by forest department. Now it's just matter of time that sooner or later some other the male will mate with these females, and the cubs that are born would ideally break the cycle of inbreeding. But I guess that's where Uma comes in again, and she studies these new cubs which are born this year or next year or the year after that, and understands how much change has happened. I think based on that, they will also continue to decide that do they want to bring in more individuals or not. So yeah.
No, this is absolutely fascinating. Last question to Uma before we take questions from the audience. Uma, I mean, there's a fantastic fascination in the public by the visual imagery of a tiger or other animals. And, everyone says, wow, this is an area of, ecology or conservation science I'd like to work in. But like every other aspect of science, um, the visual imagery is one part, but it requires a lot of hard work to get the understanding and the capabilities. And one of the ways which you have done that is to combine the study of population genetics with the prevalence in various ecosystems of these animals. Can you tell us a little bit about that and how you trace tigers in the wild and how you know about their ancestry from work in museums?
Oh yeah, yeah. So basically, I mean, when I was younger, just after my undergraduate, I was always interested in animals and I wanted to work on wildlife. and I actually started watching elephants. And I realized that, to understand, even to understand elephant ecology, using a cheat of their DNA would be really, really useful, because, you can watch animals for really long, but you can't watch them all the time, and you really don't know what exactly they're doing all the time. Also, DNA, our DNA is like a book, and it basically holds the story of our past, right? So by reading this book, we can actually understand, who is related to whom in a population, which individuals are different from each other, how different populations are related to each other, whether they're connected or isolated, and so much about ecology, as well as genetics, kind of is hidden in this DNA sequence. There's also, in the last 20 years, been a revolution in technology called next generation sequencing, which allows us to very cheaply read DNA sequence, and not just a small bit of DNA, but the entire tiger genome, which is 2,400 million letters long. So this is really great, because then we can look at all of what evolution has wrought through time. And since it's stochastic, the more data we have, the better ability to understand the past. But one of the issues is that the past that we see today in the present, lots of things happened in the past, right? But we see an averaged version of that in the present. And what would be amazing is to be able to go back in time and to actually look at what happened 100 years ago, what happened 200 years ago, what happened, why did Simlipal become isolated in the first place, right? Was it because tigers went extinct in Chhattisgarh, or was it because of habitat fragmentation in Chhattisgarh? So that's something which we've just embarked on, where we're trying to actually look at trophies or historical DNA, go back in time. The work is still ongoing, but really exciting to be able to sample and go back in the past like a time machine. Thanks for giving me the opportunity to talk about that. Yeah.
Thank you very, very much. Before I hand over to Utsav to moderate the questions, I would again urge you to get this National Geographic issue of October 2025. It's really extraordinary and stimulating. And, it's something all of us in India need to be proud of, to see what Prasenjeet has done and what research Uma has done to help understand this. And it's something which really inspires all of us to take on research in many of these kinds of areas. Over to you, Utsav.
Thanks, Vijay. Thank you so much, and thanks, Prasenjeet and Uma, for the lovely insights. It was great. So I'm just gonna. There are a bunch of questions here. Some of those, I think you have, Prasenjeet, Uma, you have touched upon them, but see if you want to take it up. So one first question is from Abhay Jain, and they have asked if there are any, like, I think this is related to what Uma was just speaking about in some bits, where they're asking, are there any survival related benefits of the mutations? Is
No, yeah, not this mutation that we are aware of, but this is something we're trying to test, but it might be very difficult to do when populations are very small. What happens is selection, which would be the force, or the evolutionary force which allows for survival benefits, becomes very weak. And the stochastic, by chance random processes become much more dominant. So it's unlikely. It seems as if all of the data is consistent with stochastic processes like drift, but it's something we're still trying to explore.
okay, all right. And Adrit has asked a question, and they're asking if would it be possible to have an all black tiger if we have two pseudo melanistic parents?
No. So basically, this is not an additive trait in the sense that we don't get a double effect or a triple effect, something like that, right?
Mm-hmm.
As far as we know, it's either pseudomelanistic or not. But I told you myself that the amount of black seems to vary. But this is not a melanistic form. For example, we have melanistic leopards. Like I said earlier, it's a pattern disruption. That is, the black stripes are not laid properly, but it's not completely black. It still retains so there's three tiger colors: orange, white, and black. It still retains orange and white colors. So it's not a color mutation, like say a white tiger has no orange, right? It's a pattern mutation. So we don't expect that we would have a completely black tiger.
Okay. All right. So one more question from Shubhodeep Datta. They have asked at one point of time we were talking about Tiger Islands, right? So they've
Mhm.
asked if there are any similar Tiger Islands elsewhere in India where we
Yes.
might have few
Yeah.
individuals who carry this mutation, but we don't know because pseudomelanism or any such visual indicators have been expressed yet. And
Excellent, excellent question. Really great question. And that's what has been really amazing about the Simlipal story is because there is a visual indicator, right? So, for
mm,
example, we've also been working for many years, almost a decade now, in Ranthambore Tiger Reserve, where also there is a lot of inbreeding. Individuals in Ranthambore are twice as inbred as those outside of Ranthambore. But there's no visual indication of this, and this is something we are still studying. We have reconstructed the pedigree, and we are actually seeing that over time, inbreeding, even in the recent 20 years, inbreeding is increasing over time. So yes, and there may be some other populations. The Sundarbans might be an ecologically isolated population because it's very unique in terms of its habitat. The southern Western Ghats, Kalkad Mundanturai Tiger Reserve, Anamalai Tiger Reserve, might also be such isolated populations. But we have the best evidence so far for Simlipal and Ranthambore.
okay, okay. And Prasenjeet, like in your experience, like when you are going to photograph or capture a particular species, then you probably like map it out also accordingly, right? Like there are certain islands or some particular places where you would get a certain species and then you would go work around there. Like how you went into like with the snow leopards when you've done your work with them, or in Mongolia when you've gone and like yeah.
Yeah. I think it's not so much about isolation, it's about finding species or individuals that I really need to photograph for telling the
Mm,
stories which are about isolation or islands. Along with along with this, I think general theme of the stories that I've worked on have something to do with islands. I've worked on sky islands, which are these mountains in the sky, and there I went to look for frogs and snakes, which are found only on these mountain tops and nowhere else in the world. Along with that, I did a story which was about this bird species called as Narcondam hornbill, which is found on one little island in Andaman and Nicobar island chain, and then this Tiger Island. So yes, a lot of research goes behind before stepping into the field. I think people have this misconception that you just go and then just wait there. And as wildlife photographer, 90% time you are just driving around in cars looking for tigers. The reality is that that's literally the 30% of the work. The remaining 70% is just sitting across computers and sitting across meetings offices, writing grants and talking to scientists and sitting across forest offices to get permits to access those places. Yeah.
all right, great. I think like this is like a very good information for all the students also over here. Because I mean, I would like to actually ask you here right now, uh, science photography and storytelling is like an option which I guess is not again spoken about so much. It is not a field that a lot of students look forward to, or like because like in the sciences also when students are studying that, and even if they're interested in ecology and everything, this option is not so readily available, right? So how would like let's say there are some high school students, or college students also undergrads, and how could they potentially like explore this pathway, something that you did, and then kind of like go into like storytelling, science communication through
That's a
photography.
great That's a great question. So when I started out, and I was a student in NCBS, I was UMA student and Vijay student at some point. Uh, but when I started doing photography, I figured out that I'm interested in telling science stories because I got into science because I was fascinated by science. I just realized that I enjoy telling science stories much more than the academic part of it. So I was like, I'll figure out a way to stay in science without doing academics, which is way more harder for me than and it's I was surrounded by Uma and Vijay and incredible scientists. So I was like, I'll find out a way to photograph science. And I think this is back in 2013, 12, 2012, 13, 14. And me, along with two, three other photographers with National Geographic, we all realized that we are interested in science and we do photography of science. So in a way, we kind of started calling ourselves science photographers. I don't think anything like that existed before. I don't think it exists even now. We just focus on science and we are photographers, so we became science photographers. And at some point, Nat Geo started calling us that, so now it is established that we are science photographers. But the process is simple. You pick up a science paper, you try and understand it in as detail as possible. Out of it, you identify specific nodes which are important to be communicated. Some nodes are important because they can put things in context for larger public. Some nodes are important because they are very pivotal into the science process of it. And some are important because they tell what the result is and what the future is. And based on that, you think of certain pictures that would help you take that story forward. The whole point is to grab people by eyeballs. And then when you have them, then they go ahead and read the scientific article. And so the students who have read this science article, they would understand, or people who have read this, they would understand a bit about this science. And some of them would be even more curious, who would read about Dr. Ruma and find the paper and would read about that paper also. So it's a good segue to get into communicating science. Now, how anyone should do this? I don't think there's a course exists. I don't think there's a path that exists. I think for every student who wants to do this, I would recommend that pick stories that are accessible to you. You are surrounded by scientists around you. Find a paper that you are excited about, and you don't have to pick only photography. You could be a writer, you could be a comedian, you could be a artist, any kind of artist. Figure out a way to communicate that science, and hopefully many of these listeners would communicate science and tell us how it is done as well in near or far future.
Thanks for that lovely answer.
Utsav,
Super
there
important.
are
Yes, yes.
two questions. One was, how can we read Professor Umar Ramakrishnan's papers? I mean, the answer is Google, find out the papers. Many of them are online. You should be able to download them and read it. But if you can't get hold of them, drop her an email and she'll send you the PDF. So that was one question. And the other one, which I thought was interesting, was yeah, yeah. For animals in the wild, how do we extract their genetic material, especially in the case of protected species such as tigers, because it's illegal and bad to hunt them down to study them. So Uma has a solution for that. It's very simple. And Uma, can you tell us about it?
Yeah, so basically, especially in the field, in protected areas, we work with non-invasive samples. So basically, we either work with fecal samples, where, basically the feces passes through the intestine, and as it does, it pulls off epithelial cells from the intestine. And so basically, there are tiger cells, usually in that mucus kind of shiny layer of the feces or the tiger poop, and you can basically take a swab and just kind of rub it on the surface of the poop, put it into a buffer, and bring it back to the lab and extract DNA from it. Now with poop you get tiger DNA, but also DNA from what it ate, bacteria and so on, but you do get tiger DNA, and you can design specific experiments called PCR based experiments and specific primers which pull out parts of the tiger genome. On the other hand, you can also sequence whole tiger genomes from non-invasive samples. And we've been doing this very successfully with shed hair, because shed hair is a kind of a purer source of just tiger DNA. So basically, you watch animals, or you use, right, what we've done just recently in Simlipal. We use camera traps to identify where animals are moving, just as Prasenjit did. In fact, Prasenjit helped us out the first week we were in field. He was leaving, and he told us where to look out for which individuals and so on. And so basically, the amazing thing about animals is they're really creatures of habit. They follow the same route, and you can predict 7 days from now this individual will move through here, and that's so amazing. I mean, from the outside, it seems like it's so mysterious, but they actually, once you start watching them, they follow these patterns, like, of time, in time. So then you can basically either watch them, or, figure out how they're moving and collect shed hair. And then basically again bring it back to the lab, extract DNA, and in this case, you can sequence whole genomes. Now, whatever work we do, because tigers are listed on the Wildlife Protection Act under Schedule 1. That means they're protected everywhere they are, inside a park, outside a park, inside a zoo, anywhere, right? So we have to get permissions from the Forest Department to both enter the protected area as well as collect these samples. So everywhere we work with permissions. And in fact, you're right. The interesting thing about the Wildlife Protection Act is sampling any kind of tissue is akin to hunting. So there's no particular like mention in the act of research based sampling. So it's very interesting that we still are, in some sense, taking or extracting something from the natural habitat. So we do require permissions for all the work we do.
Uma, on the same note, there's a question. This is by any anonymous attendee. They're asking if you can differentiate tiger DNA from prey DNA via fecal samples, like, or do you run
Yes.
it with against a pre-existing existing sequence tiger genome, or and
Yes, yeah.
how
So
does it?
basically, every, like we just discussed, right? Every species has a unique sequence. Every individual has a unique sequence, right? So basically, we design primers, which are pieces of little bits of DNA which stick to your, the DNA you've extracted, and then you amplify it, or you read and make many copies, say between these two, right? But the thing is that those sticky things will only stick to tiger DNA. You design them, you engineer them, you read, you see tiger DNA, you map against it like prey DNA, and you see, oh, these parts are similar, but these parts are really different, right? So you design your primer, your experiment, such that you're pulling out the different parts, right? So you can, and then you're right, Utsav, there's this huge database called GenBank, which has all DNA that everyone has ever sequenced. And now we have going to be equivalent in India as well,
It's
hosted
publicly
by the Department
available,
of
like the
Biotechnology.
this
So you can
repository, this
actually,
data.
yeah, blast
Oh,
it, like throw it at
okay.
that massive mega set of DNA, and it returns results using like a
Wow.
bioinformatic approach called BLAST, looking for similarity in sequence, and you can figure out what DNA you have. But
Mm-hmm.
as you said, we can also specifically amplify prey DNA. So we can also figure out what the tiger has eaten or the leopard has eaten. And that's so cool because we can actually look at, diet and things like that as well.
Yeah, that's amazing. Okay, one question we have from Arish Azmat for Prasenjeet. They're asking about what camera gear and lenses do you usually rely on for your photography, and do you prefer natural lighting, and how do you deal with harsh lighting conditions in outdoors?
That's
So,
a
if you
great
could share.
question. I don't have a camera brand allegiance because there are times when I'm photographing macro stuff and some other brand works, and there are times when I'm photographing huge plants and some other brand works. There are times when I'm filming and some other brand works. I do have a lot of Nikon gear. Some of my camera traps are built out of Nikon. So they are all different cameras, right? From something basic to something as advanced as Z8. And I do prefer natural light, but there are times when I'm photographing at night, just like these camera traps where these tigers are not very easy to find, and setting up camera traps, hoping to get their photos at night. That's when I had to rely on artificial lighting, which is different flashes, and essentially, I rarely photograph with equipment that is off the shelf. Usually, we build new contraptions for different species or different stories or different projects, and use that to create pictures which are Nat Geo worthy.
Okay,
I hope that
great.
answers your question, Arish.
Super. Okay, we'll take one more question before concluding the webinar. This is from an anonymous attendee. They are asking if the tigers were once normal, did a random mutation transfer across generations, and then the pseudo-melanistic population come into being? And is this the base for introducing new tigers? Would any of you have some
Um, So I'll
insight?
I'll try to answer that. So
Huh.
basically, what we think happened in Simlipal was what's called a population bottleneck. That is, there were some individuals, and then for some reason, either it progressively got fewer, or there has to have been some point where there were very few individuals, maybe two, three, four, five, something like that. And one of those individuals had to have had this mutation, right? And then subsequently, because there were such few individuals, there was inbreeding and mating between all of these. They're all related anyway now because there's only four of them or something, right? So as they mate with each other, this pseudomelanistic mutation becomes visible in the phenotype. I hope that answers your question. But there are historical records of pseudomelanistic tigers in other places, not very far off, maybe a few other places. I mean, it's not clear from the historical records whether they're pseudomelanistic, they're kind of roughly called black. So it could be that this mutation was there elsewhere. But today, as far as we know, so we did look at about 400 tiger samples across, different places. We could not find even a heterozygote of the pseudomelanic mutation. That is, an individual that was a carrier. We didn't have even that that we saw. So yeah.
Okay. Okay. Great. Thank you for answering all of these questions. There are some remaining, but I think we are running out of time now. But thanks again, Prasenjeet and Uma, for this lovely discussion, and Vijay for moderating it so well. For any school educators over here, like school educators and any students, by the way, who are in high school, I would just like to mention that the Lodha Genius Ashoka University program, the applications are also opening in another 9 days
Mhm.
from 1st of December. So please feel free to apply, go to the website, check out this program. And we have like amazing folks like Prasenjeet and Uma who contribute to the program also. And there's a lot of interdisciplinarity that we focus on through this program. And we also hope through such discussions your students and some other folks also get like inspired to take up such to know more about such fields, right? And to basically maybe engage in it in some ways, and you have something to take back from here, from this discussion or from the program. But thank you so much everyone for joining this discussion. It was lovely having everyone. Thanks Prasenjeet, thanks Uma, and thanks Vijay.
Thank
Thank
you.
you. Thank you.
Thank you.
And
See you
Thank
all
you,
soon.
everyone.
Bye.
Thank
congratulations,
you.
everyone. Thank you.
Thank you. Take care.
Bye.